Hello, welcome back. Here we have the video on the answer key for the question video code, KUBotS1A1. Hope this too helps. Thank you all for the support. Botany, answer key, question video code, KUBotS1A1. Answer video code, KUBotS1A1. QP1. 1 mark questions. 1. Supportive cells that aid in the transport of nutrients and sugars in plants. They provide metabolic support and maintain cell integrity. 2. The arrangement of ovules within the ovary of a flower. It influences seed and fruit development. Types include marginal, basal, axial, and free central placentation. 3. Large, specialized cells in grass leaves that control leaf rolling. They regulate water uptake and help in the plant's response to environmental conditions. 4. Fusion of male and female gametes during fertilization, leading to the formation of a zygote. 5. The study of pollen grains, their structure, development, and characteristics. It includes their role in plant reproduction and forensic applications. 6. Nectar functions to attract pollinators and serves as a reward for them, promoting pollination and enhancing plant reproduction. It also provides energy for pollinators in the form of sugars. 7. Apical cell theory, proposed by Matthias Schleiden and Theodor Schwann in the mid-19th century, suggests that the growth of plants occurs primarily through the division of apical cells at the tips of roots and shoots. 8. Plasmodesmata, microscopic channels in plant cell walls connecting adjacent cells, facilitate the exchange of ions, molecules, and signals between plant cells, contributing to coordinated growth and development. 9. Phelogen, also known as the cork cambium, it's a meristematic tissue in plants, responsible for the production of cork cells, contributing to the outer protective layer in stems and roots. 10. Triple fusion, occurs during the fertilization of flowering plants, involves the fusion of 3 nuclei, 1 sperm nucleus with 2 polar nuclei in the embryo sac, leading to the formation of endosperm in seeds. 2 mark questions. 11. Applications of Palynology in modern classification, link in the description box. 12. Porogamy versus cholezogamy. Porogamy. Fertilization occurs through an opening or pore in the ovule, often near the microcoel, facilitating the entry of the male gametes. Cholezogamy. Fertilization occurs at the chalaza, the basal part of the ovule, without the involvement of an opening or microcoel. Porogamy examples. Orchids exhibit porogamy, where pollen tubes grow through a small opening to reach the ovule. Cholezogamy examples. Pine trees demonstrate cholezogamy, with pollen tubes growing towards the chalaza for fertilization. 13. Hydothodes and their functions. Definition. Hydothodes are specialized structures in plant leaves that excrete water in the form of droplets. Water regulation. Hydothodes help regulate water balance in plants by releasing excess water, preventing waterlogging and maintaining turgor pressure. Mineral excretion. They aid in excreting minerals dissolved in water, preventing their accumulation and potential toxicity. Cooling mechanism. Hydothodes contribute to leaf cooling by releasing water droplets through a process known as guttation. Pathogen defense. Hydothodes can flush away potential pathogens, acting as a defense mechanism for the plant. 14. Heartwood versus sapwood. Heartwood. It is the inner, non-living, darker part of the tree trunk, characterized by its higher density and resistance to decay. Sapwood. It is the outer, living, lighter-colored wood responsible for transporting water and nutrients, usually found closer to the bark. Heartwood functions. Provides structural support, stores waste materials, and contributes to the overall strength of the tree. Sapwood. Functions. Conducts water and nutrients, participates in the tree's metabolic activities, and serves as the tree's primary transportation system. 15. Pits and their classification. Definition. Pits are small areas of secondary cell wall that lack lignin, allowing for communication and transportation of water between adjacent plant cells. Simple pits. Basic pit structure with a single opening between 2 cells, facilitating direct water movement. Bordered pits. Contains additional cell wall material forming a border around the pit, influencing the flow of water and preventing air embolisms. Perforation pits. Found in vessel elements, they are larger openings that enhance water conductivity between vessels. Pitted parenchyma. Specialized parenchyma cells with numerous pits, promoting efficient water movement within plant tissues. 16. Interfascicular cambium versus intrafascicular cambium. Location. Interfascicular cambium is found between the vascular bundles in dicot stems. Intrafascicular cambium is present within the vascular bundles in dicot stems. Origin. Interfascicular cambium originates from the interfascicular parenchyma. Intrafascicular cambium originates from the procambium during primary growth. Function. Interfascicular cambium contributes to the secondary growth of the stem by producing secondary xylem and phloem. Intrafascicular cambium also plays a role in secondary growth, forming additional vascular tissues within the bundles. Connection. Interfascicular cambium connects the vascular cambium of adjacent vascular bundles. Intrafascicular cambium connects with the interfascicular cambium to form a continuous ring during secondary growth. 17. Plants yielding fibers of economic importance. Cotton, Gossypium spp. Flax, Linum usitatissimum. 18. Functions of cambium in monocots. Vascular tissue formation. Cambium in monocots contributes to the formation of secondary vascular tissues, aiding in the increase of stem diameter. Wound healing. It plays a role in healing wounds and injuries in the stem by generating new cells. Transport of nutrients. Cambium facilitates the transport of nutrients and water between different parts of the plant. Structural support. The secondary tissues produced by cambium provide structural support, contributing to the overall strength of the plant. 19. Cystoliths. Defined link in the description box. 20. Palynicoma and types. Definition. Palynicoma is a type of plant tissue that provides structural support, particularly in growing regions. Types. Angular palynicoma. Cells with thickened corners. Lacunar palynicoma. Cells with thickenings at the cell corners and in the tangential walls. Annular palynicoma. Cells with thickenings in a ring-like pattern. Tangential palynicoma. Cells with thickenings mainly in the tangential walls. 21. P. tapetum and C. tapetum. P. tapetum, post-meiotic tapetum. Develops after meiosis and supports the maturation of pollen. Provides nutrients to developing pollen grains. C. tapetum, callose tapetum. Forms during meiosis and helps in the release of pollen. Involved in the formation and degradation of callose, a substance crucial for pollen wall development. 22. Megasporogenesis. Essay explained. You can watch and add even more points. Link in the description. Four-mark questions. 23. Structure and development of dicot and monocot embryos. View and add more points for your exam. Link in the description. 24. Fertilization is the process of fusion between male and female gametes, sperm and egg cells, to form a zygote, which develops into a new individual. Barriers to fertilization include mechanical barriers, physical obstacles, such as the pollen wall, which prevents sperm from directly reaching the egg. Self-incompatibility (SI) barriers. Inability of a hermaphrodite plant producing functional gametes to effect fertilization upon self-pollination. Is a prefertilization barrier, mainly to prevent self-pollination. Intraspecific. Within the species. Interspecific. Between species. Categories. Gametophytic and sporophytic self-incompatibility. 25. Endosperm and their types. Link in the description. 26. Apical meristem theories. Link in the description box. 27. Secretory tissue is a type of specialized plant tissue that primarily functions in the production and secretion of substances such as enzymes, pigments, and other organic compounds. These substances play crucial roles in various plant processes, including defense mechanisms, growth, and reproduction. Secretory tissues are classified into two main types: latocyphers and non-latocyphers. Latocyphers are specialized secretory tissues found in some plants, particularly in the family Euphorbiaceae. These tissues produce and secrete lactones, which are a type of organic compound. Latocyphers are unique because they contain a unique cell structure called a lactiferous cell, which is responsible for the synthesis and storage of lactones. Latocyphers are further classified into two types: true latocyphers. True latecyphers are found in plants such as Euphorbia, a genus that includes plants like the poison spurge and the crown of thorns. False latecyphers. False latecyphers, also known as pseudo-latecyphers, are found in plants like the milkweed Asclepias and the castor oil plant Ricinus communis. Non-latecyphers are secretory tissues that do not produce lactones. Instead, they secrete other types of organic compounds, such as enzymes, pigments, and alkaloids. Non-latecyphers can be found in various plant tissues, such as glandular trichomes, hair-like structures, and parenchyma cells. Some examples of non-latecyphers secretory tissues include mucilage-secreting cells. These cells secrete a slimy, gel-like substance called mucilage, which helps in plant hydration and provides protection against desiccation. Oil glands. Oil glands are specialized secretory cells that produce and store lipids, such as essential oils and resins. These substances are often used for defense against herbivores and pathogens. Nectary glands. Nectary glands are found in flowers and produce nectar, a sweet liquid that attracts pollinators. 28. Stomata are microscopic pores on the surface of plant leaves, responsible for gas exchange and transpiration. Types of stomata. There are different types of stomata and they are mainly classified based on their number and characteristics of the surrounding subsidiary cells. Listed below are the different types of stomata. Diagrammatic representation is as follows. Anemocytic stomata. They are surrounded by epidermal cells, which have a fixed shape and size. The stomata appear to be embedded in epidermal cells. There is no definite number and arrangement of cells surrounding the stomata. Anisocytic stomata. Stomata are surrounded by 3 subsidiary cells having unequal sizes, one is smaller compared to the other 2. Diacytic stomata. The stomata are surrounded by a pair of subsidiary cells that are perpendicular to the guard cell. Parasitic stomata. The stomata are continuously surrounded by 2 subsidiaries, which are arranged parallel to the stomatal pore in the guard cells. Gramineous stomata. Each stoma possesses 2 guard cells, which are shaped like dumbbells. The subsidiary cells are parallel to the guard cells. The guard cells are found narrow in the middle and wider at the ends. 29. The anatomy of a dicot leaf consists of several layers and structures, including epidermis, cuticle, mesophyll and vascular tissue network. The following diagram shows the anatomy of a dicot leaf. Cuticle. A waxy layer covering the epidermis, which prevents water loss and protects the leaf from pathogens and environmental stress. Adaxial epidermis. The outermost layer of the leaf, responsible for protecting the leaf from external factors and regulating gas exchange. Palisade mesophyll. A layer of closely packed, elongated cells located below the epidermis, primarily responsible for photosynthesis. Spongy mesophyll. A loosely packed layer of cells located between the palisade mesophyll and the lower epidermis, which also plays a role in photosynthesis and gas exchange. Vascular tissue. A network of xylem and phloem tissue responsible for transporting water, minerals, and sugars throughout the plant. Abaxial epidermis. The innermost layer of the leaf, which is typically thinner and lacks the waxy cuticle found in the upper epidermis. 30. Anomalous secondary growth. Explained link in description. Watch the video. There are more points which you can add up to your knowledge pack. 31. You can also get more points from the video, Secondary Growth, Explained. Link in the description. Extrastellar secondary growth refers to the process by which plants increase their diameter and girth without forming new cells in the stellar region. This growth occurs in plants with secondary thickening, such as trees and some shrubs. The process is essential for the development and support of larger plants, enabling them to store more nutrients and water, and providing structural stability. The vascular cambium is a thin layer of meristematic tissue located between the primary xylem and primary phloem of plants. It is responsible for producing new xylem and phloem cells as the plant grows. During secondary growth, the vascular cambium divides to create new xylem cells, secondary xylem, on the inside and new phloem cells, secondary phloem, on the outside. As these cells mature, they form new layers of wood and bark, which contribute to the overall thickness of the plant stem. This process allows the plant to increase in girth while maintaining its original height. The cork cambium, also known as the phellogen, is a layer of meristematic cells located just below the outermost layer of the plant, called the epidermis. During secondary growth, the cork cambium divides to produce new cork cells, phellum, on the inside and suberized parenchyma cells, phelloderm, on the outside. These new cells differentiate and become part of the protective outer bark layer. The cork cambium's activity also plays a crucial role in wound repair, as it produces cork cells to cover damaged areas, preventing water loss and pathogen invasion. 15 mark questions. 32. You can refer to the video, Permanent Tissues, notes for the answer. Link in description. Just watch and add up points will certainly increase the size of your knowledge basket. Diagram of complex tissue. 33. Anomalous secondary growth in Dracaena explained. Link in the description box. 34. Meristematic tissues are a group of cells in plants that are responsible for the growth and development of the plant. They are characterized by their ability to divide and differentiate into various types of cells, allowing for the continuous growth and renewal of plant tissues. The primary characteristic of meristematic tissues is their indifferentiated nature, meaning that the cells have not yet specialized into specific cell types. Meristematic tissues are found in areas of the plant where growth occurs, such as the tips of roots and shoots, as well as in the vascular cambium and cork cambium. These tissues are responsible for primary and secondary growth in plants, allowing for increases in length and girth. Meristematic cells have a high rate of cell division, which contributes to the rapid growth and development of plant tissues. Types of meristematic tissue. The meristematic tissue is of the following types. Meristematic tissue on the basis of origin. Promeristem, the earliest and youngest meristematic tissue. It originates from the embryo. Primary meristem, arises from the promeristem. It is found in the root and the shoot tips. It arises from the promeristem. Cells divide actively. It is found below the promeristem and forms the permanent tissue. Secondary meristem, it originates from the primary meristem. The permanent tissue forms from the secondary meristem. Meristematic tissue on the basis of position. Apical meristem, these are present at the tips of the roots and shoots and help in the increase of the height of the plants. Various cell divisions facilitate the growth of the cells in the roots and shoots and help in cellular enlargement. Apical meristem is divided into promeristem zone, which contains actively dividing cells, and the meristematic zone, which contains protoderm, procambium and ground meristem. Intercalary meristem, it is located in the leaves and internodes at the intercalary position. These help to increase the length of the internode. It is found in grass, monocots and pines. It is a part of apical meristem and adds to the height of the plant. Lateral meristem, it is located in the stems and roots on the lateral side. It increases the thickness of the plant. Vascular cambium and cork cambium are the two lateral meristems. These divide preclinically or radially and give rise to secondary permanent tissues. Meristematic tissue on the basis of function. Protoderm, it is the outermost plant tissue and forms the epidermis. It protects the plants from any mechanical shocks. Procambium, it is the innermost tissue and gives rise to xylem and phloem. It helps in the transport of water and nutrients to different parts of the plant. Ground meristem, the cells are large with thick walls. It forms the cortex, pericycle and pith. The meristematic tissue is usually found in the apices of the root systems and the shoots and is in a continuous state of division. 35. You can watch the link for more knowledge and answer for this question. QP2. 1 mark questions. 1. Pericycle. 2. Filiform apparatus. 3. Blind pith refers to the central region of certain plant stems that lacks vascular tissue. This type of pith is characterized by the absence of intercellular spaces and is commonly found in monocotyledonous plants. 4. Perisperm is a nutritive tissue found in some seeds, derived from the nucellus or integument of the ovule. It serves as a source of nutrients for the developing embryo within the seed. 5. Tissue is a group of cells that are similar in structure and perform a specific function within an organism. In plants, tissues can include meristematic tissue, ground tissue, and vascular tissue, each with its own specialized role in growth, support, and transport. 6. Xenogamy is a type of cross-pollination in plants where pollen is transferred from the flower of one plant to the stigma of a flower on a different plant. This process promotes genetic diversity within plant populations. 7. Anemophily refers to the pollination of flowers by wind. In anemophilous plants, Pollen is dispersed by air currents to reach female reproductive structures, often without the need for nectar or showy petals to attract pollinators. 8. Root epidermis. 9. False. 10. Callose is a carbohydrate polymer that forms at various stages of plant development, including during cell wall formation and in response to stress or injury. It plays roles in cell signaling, defense against pathogens, and regulation of plasmodesmata. 2 mark questions. 11. A stele refers to the central cylinder of vascular tissue in the stem and roots of vascular plants. The stele is composed of xylem and phloem tissues, which are responsible for the transport of water, nutrients, and sugars throughout the plant. The arrangement of the vascular tissues within the stele can vary among different plant species, leading to different types of stels such as protostele, siphonostele, and eustele. 12. Both mollicophily and chiropterophily are examples of specialized pollination mechanisms where certain animals play a crucial role in transferring pollen between flowers. Mollicophily refers to the pollination of flowers by snails or slugs of phylum Mollusca. Chiropterophily refers to the pollination of flowers by bats. 13. You can grab more for this topic from the video, Development of Male Gametophyte, link in description. 14. Concentric vascular bundles are a type of arrangement of vascular tissues in plant stems. In this arrangement, the xylem and phloem tissues are arranged in concentric rings, with xylem located towards the center and phloem surrounding it. This organization is characteristic of monocotyledonous plants and is in contrast to the radial arrangement found in dicotyledonous plants. 15. Crassinicellate ovules have a thick nucellus, which is the central part of the ovule containing the embryo sac. In contrast, tenuinucellate ovules have a thin nucellus. This difference in nucellus thickness is one of the distinguishing features between these two types of ovules. 16. Exarch xylem refers to xylem in which the protoxylem, the first formed xylem, is located towards the outside of the xylem strand. Found mostly in root structures. Endarch xylem refers to xylem in which protoxylem is located towards the inside. Found mostly in stem structures. These terms are used to describe the arrangement of primary xylem elements in plant stem and roots. 17. Mesogamy. Mesogamy is a term used to describe fertilization that occurs between gametes from different individuals within a population or species. This process promotes genetic diversity within offspring by combining genetic material from distinct parental sources. Mesogamy plays a crucial role in maintaining genetic variability within plant populations, which can enhance their ability to adapt to changing environmental conditions. Angiosperm. Angiosperm is a nutritive tissue found in seeds of flowering plants that provides nourishment to the developing embryo. It is formed as a result of double fertilization, where one sperm cell fuses with two polar nuclei in the embryo sac to form triploid endosperm tissue. Endosperm serves as a source of energy and nutrients for the growing embryo during seed germination and early seedling development. 18. The tapetum is a layer of nutritive cells found within the anther of flowering plants that supports pollen development and maturation. There are several types of tapetum based on their cellular characteristics, including secretory tapetum, which produces substances for pollen wall formation, plasmodial tapetum, with multinucleate protoplasts, amoeboid tapetum, with amoeboid-like cells, and glandular tapetum, which secretes substances important for pollen development. Each type plays a distinct role in supporting pollen grain formation. 19. Lime trees, Citrus aurantifolia, possess specialized glands known as oil glands or oil cells located within their leaves, flowers, fruit rinds, and other plant parts. These glands secrete essential oils that contribute to the characteristic aroma and flavor associated with limes. The oils produced by these glands contain aromatic compounds that are valued for their culinary uses as well as for their potential medicinal properties. 20. Pollination is the process by which pollen is transferred from the male reproductive organs of a flower to the female reproductive organs, thereby enabling fertilization and the production of seeds. This transfer can occur within the same flower, self-pollination, or between different flowers of the same species, cross-pollination. The advantages of cross-pollination include genetic diversity. Cross-pollination promotes genetic diversity by combining genetic material from different individuals, leading to offspring with varied traits and increased adaptability to changing environmental conditions. Disease resistance. Cross-pollination can result in offspring with enhanced resistance to diseases and pests, as it introduces new genetic combinations that may confer resistance traits. Improved fitness. Cross-pollination can lead to increased vigor and overall fitness in plant populations, as it allows for the exchange of beneficial genetic traits. Hybrid vigor. Cross-pollination can give rise to hybrid vigor, where the offspring exhibit superior qualities compared to their parents, such as increased growth rate or yield. 21. Casparian stripes are bands of impermeable lignin and suberin-containing material found in the endodermal cells of plant roots. These strips encircle each cell and create a barrier that prevents water and solutes from freely passing between cells. Casparian stripes play a crucial role in regulating the movement of water and nutrients into the vascular system of plants, ensuring selective uptake and preventing the entry of harmful substances. 22. Tylosis refers to the formation of outgrowths or projections from living cells into adjacent non-living cells, particularly in woody plants. These outgrowths are often formed in response to injury or infection, serving as a defense mechanism to block the spread of pathogens or seal off damaged areas. Tylosis formation involves the deposition of substances such as gums, resins, and other compounds that help to obstruct the passage of pathogens and promote wound healing in plants. 4-mark questions. 23. Repeat. View Case No. 28 of the previous paper. 24. You can grab more for this topic from the video, Structure of Mature Anther link in description. 25. Dicot leaf. Dicot plants have leaves that are relatively smaller and broader than monocot plants. Stomata in dicot leaves are kidney-shaped. Dicot leaves are hypostomatic, which means stomata are present on the lower surface of the leaf. Differentiated into two parts, upper palisade and lower spongy mesophyll. Dorsoventral orientation. Monocot leaf. Monocot plants have leaves that are characteristically longer and slender. Stomata in monocot leaves are dumbbell-shaped. Monocot leaves are amphistomatic, which means the stomata are present on lower as well as the upper surface. No such differentiation in a monocot leaf. Isobilateral orientation. 26. Anomalous secondary growth in Dracaena explained. You can grab more points to answer from the video link in description. 27. Stem. The epidermis of the stem is cutinized. The epidermis of the young stem possesses stomata. Stem epidermis contains multicellular trichomes. Stem possesses a narrow cortex. The hypodermis is present in the stem. The stem contains differentiated cortex as the outer, middle, and inner cortex. Endarch xylem, and are present in stems and roots respectively. Pericycle may single or multi-layered in stems. Root. Root epidermis is not cutinized. Root epidermis does not have stomata. The root contains unicellular hairs. But the root cortex is broad. Hypodermis absent in root, but the root contains an undifferentiated uniform cortex. Root has distinct endodermis. Exarch xylem. The pericycle is single-layered in the root. 28. Double fertilization is a unique process that occurs in the life cycle of plants belonging to the Angiosperms group, also known as flowering plants. This process involves the fusion of two sperm nuclei with two different female gametes, resulting in the formation of a zygote and an endosperm. Double fertilization is crucial for the development and survival of the seed and embryo in flowering plants. Inside the ovule, there are two female gametes, the egg cell and the central cell. The sperm cells from the pollen grain enter the ovule through the pollen tube. One sperm cell fuses with the egg cell, resulting in the formation of a zygote, while the other sperm cell fuses with the central cell, leading to the formation of the endosperm. 29. You can get an answer to this from the essay explained in the video, Ultrastructure of Cell Wall link in description. 30. The key difference between anatropous and orthotropous ovule is that anatropous is the most common ovule orientation in angiosperms, in which the ovule is completely inverted in 180 degrees so that the micropyle and hilum come closer to each other. Meanwhile, orthotropous is an ovule orientation in which the micropyle lies directly in line with the hilum, i.e. straight. Following diagram shows the detailed vision for you. 15-mark questions. 31. Dicot. The dicot stem is solid in most of the cases. The hypodermis is formed of parenchyma fibers which are often green in color. The internal tissues are arranged in concentric layers. The ground tissue is differentiated as endodermis, cortex, pericycle, medullary rays, pith, etc. The vascular bundles are formed as broken rings. Phloem parenchyma is present. Pith is well developed. Epidermal hair may or may not exist. Vascular bundles are less in number and are of uniform size. The dicot stem does not have a bundle sheath on the outside of a vascular bundle. The dicot stems have trichomes. The vascular bundles always remain open, due to the presence of cambium within phloem and xylem. Dicot stem can feature secondary growth as a result of secondary vascular tissues and periderm formation. Vessels are of a polygonal shape and are arranged in rows or chains. Usually, vascular tissues stop functioning when they get old. New vascular tissues replace the old ones. Monocot. The monocot stem is usually hollow at the center. The hypodermis is made of sclerenchyma fibers and they are not green. There is no concentric arrangement of tissues. The ground tissue is the same and is composed of a mass of similar cells. The vascular bundles are scattered irregularly around the ground tissue. Phloem parenchyma is absent. Pith is not as well developed in monocots, usually absent in most. Presence of epidermal hair. There are numerous vascular bundles of different sizes. The monocot stem has a sclerenchymates bundle sheath on the outside of a vascular bundle. The monocot stems do not have trichomes. The vascular bundles are closed. No secondary growth is witnessed in case of monocots. Vessels are rounded or oval and are arranged in a Y-shaped formation. Vascular tissues remain the same throughout the plant's life cycle. 32. Find the video of anomalous secondary growth in dicot stem essay, Boerhaavia, explained link in the description. 33. Kindly find the video of megasporogenesis explained for the essay link in the description. 34. Repeat. View K's No 34 of the previous question paper. Thank you. This is a recommended video. We are glad to see and work according to your recommendations, suggestions and all we get. So kindly like, share, comment and subscribe us for more videos.